There's a lot that goes into being an animal or what constitutes an animal. I've got a big Galapagos tortoise here that I just took a picture of while I was in the Galapagos Islands. So today we're going to talk about the anatomy and physiology of organisms. So we're really looking at animals at the organismal level. There's so many different things that make them unique individual species, but also there's a lot of things which animals have in common. So when we talk about anatomy and physiology, we are going to look at these processes or these structures and functions at different levels. The first level is, or the smallest level, is at the cytoplasmic level, where we're looking at differences within the cell. So I have a little cross-section of a cell down here. Right? And you can see all the little organelles, and there's DNA in the nucleus, and there's genetic and molecular changes which are taking place, which then interact with each other within that cell. You can look at the cellular interactions. So in a multicellular organism, you're going to have specialization of different types of cell, and each cells then can be different, slightly different in their function. Colonial organisms are those that can create these colonies, but each individual cell is pretty much exactly the same and can reproduce on its own. Tissue then is looking at cells with similar functions that are aggregated together. So you have connective tissue and epithelial tissue. We'll talk about some of those later. Then an organ is another level of organization where you have a similar function. A heart is a gathering of different types of tissue that are all contracting to pump blood through the organism's circulatory system. And finally, systems work together then for specific functions. Like I said, circulatory system is made of blood vessels, the heart, blood, tissues, organs, cells, all of these things working together. There are 11 different organ systems in animals, including nervous system, digestive system, immune system, etc. All right, so then looking at anatomy, we are going to try and understand what are the different parts of an animal, and it helps us to understand then how to orient our organisms, and that can depend on what their body plans are. The first way we can look at body plans is in their symmetry. So some can have no real symmetry, like a sponge. It's asymmetrical, it's not round, it's just kind of growing and not random, but not in a very organized way. Radial symmetry is then these radial shaped animals, not circular, but at a certain point they looking from above, they look circular, but they're more columnar. And then bilateral symmetry is where they have left and right halves, but their front and back and top and bottom are all different. So they are only similar on one plane. All right, bilateral symmetrical animals also have certain terms that we need to use to help define their orientation. Bilateral symmetry generally means they have a head and a tail and then some appendages, but not always the case, but generally that's how we speak of it. And so that would be cephalization, where you have the formation of a head. To help us orient then different parts, we can talk about these terms of location. You've got this horse over here. You can see the anterior towards the head, posterior towards the tail, ventral underneath, dorsal above, so towards the back, dorsal towards the belly, ventral. Medial is towards the middle, which isn't pictured there, and lateral towards the side. Distal means towards a point of attachment, and distal means away from the point of attachment, and proximal means towards the point of attachment. So you see that on the horse's leg. The knee is distal to the shoulder joint, or the toe is distal to the knee. And then we can also structure different ways to look at an organism based on body planes. You have mid-sagittal plane, which cuts it into left and right halves, down the midline. You have the frontal plane, which cuts it into front and back, right? The head and the rear. And then you have the transverse plane, which also, well, I guess it depends on the type of organism, whether it's quadruped or biped, but transverse plane then cuts like a section through your belly, like slicing you in like a salami. So that separates your head from your feet. All right, those are then the three different ways, or ways that we can then talk about different body parts. All right, development. So this has some specific anatomy, and it is really talking about how organisms go from single-celled organisms, which is a zygote, the fertilized egg, to then dividing into a specialized multicellular organism. So the first steps of cell division are called cleavage, and there are two different types. You can have radial cleavage, which has a mosaic of development, which means that each individual cell has already specialized at least a little bit. And so when you remove that cell, it can't develop independently to an organism. And then you can see radial cleavage also forms the cleavage furrows in line with others. Whereas spiral cleavage, those cleavage furrows, those lines between them, are offset by 90 degrees, so they don't line up in their cellular divisions. Spiral cleavage also has regulative development, where one cell then can become a whole new organism. Hasn't differentiated into something else at that point. All right, cleavage then leads to multiple specializations of tissue eventually. But before it gets to that, it'll form a hollow ball of cells called a blastula. And then it will go through gastrulation, where it's starting to differentiate different parts. And those different parts are called germ layers, right? So these are the layers which eventually become different types of organs: the ectoderm, the endoderm, and the mesoderm. So you can see that down here on this gastrula. You have an outer ectoderm, an inner endoderm, and then a middle mesoderm. And then I also want you to notice the blastocele. The blastocele then is the inner part of the blastula, which is the space. And those will then eventually form the coelom, or the body cavity. You can have an acoelomate, so you see that up here in the acoelomate. The mesoderm takes up the whole coelom, so there is no body cavity. There are different organisms with that. And you have a pseudocoelomate, where the mesoderm lines the outer edge of the blastocele. So you can see that there. The mesoderm in green is the outer part, and then the endoderm in red is the inner part, and then the pseudocoelom then is that blastocele. And the coelomate, which has the body cavity form inside the mesoderm. So you see the green mesoderm is completely lining the whole body cavity. All right, so this is just to show the ectoderm, mesoderm, and endoderm become different types of tissues. This is in humans as a reference point. So ectoderm becomes skin cells, neurons, and pigment cells, whereas mesoderm becomes muscle tissue, parts of the kidney, smooth muscle, and red blood cells, and then endoderm becomes lungs, other organs, pancreas, so on and so forth. All right, so we can then categorize these different processes into major divisions of development. Protostomes have spiral cleavage and a mosaic embryo, those two go hand in hand as before. They also have the original opening, which is called the blastopore. You see that below there. When it develops, that blastopore becomes the mouth, and the coelom forms by splitting the mesoderm. Whereas in deuterostomes, they have radial cleavage, a regulative embryo. The blastopore becomes the anus, and then the other side becomes the mouth. And a coelom forms by outpocketing. Okay, so you can see that outpocketing of the mesoderm there, as opposed to it splitting in protostomes. All right, we're going to switch and now talk about different types of tissues. If you remember, these are groups of cells with a common function. And histology is looking at the study of tissues, which generally is done under the microscope. There are 4 kinds of tissue. We're going to go over them in a little more detail. Epithelial, connective, muscular, and nervous. We'll start with the epithelial tissue. This covers external or internal surfaces. So the outer part of your skin, the external, that's going to be an epithelial lining. Or the inner part of, let's say, your digestive system can be completely covered with epithelial cells. So they function in protection, secretion, and absorption, those things that you would expect on the outer and inner parts of your body. They're supported by a basement membrane. That's what holds it all together. And in that basement membrane, it has a extracellular matrix containing collagen. And collagen is a very abundant structural protein found in animals. It can be in simple single layers. You can see this up here. This pseudostratified columnar epithelia is just one layer of cells. Or they can be in layered forms, which would be called stratified. We already gave some examples. So there's an example of some skin here. The epidermis, it doesn't show it, but that's a layered form of epidermal epithelial cells. All right, connective tissue, this functions for support. It connects 2 different things. It also can bind tissues and keep them stabilized. There's largely a fluid matrix with few cells and a varying amount of supportive fibers. Loose connective tissue contains fibers and that are both can be fixed or wandering cells. For example, areolar tissue, I don't have an example of that, but areolar tissue is the tissue surrounding your skin and blood vessels, and it's supportive, cushions, and it can also expand when things inflame. Dense connective tissue contains densely packed fibers, such as in tendons and ligaments, so they're all kind of running in the same direction in tendons and ligaments, and it can then tie a muscle to a muscle, muscle to a bone or bone to a bone, and hold those in place. Cartilage, loosely packed cells with fibers embedded in mostly gel-like matrix. Your ear is made of collagen, the end of your nose, very flexible material. Bone, of course, you know what bone is. It's very hard and is for providing a scaffolding for muscles to attach to, forms a skeleton. And vascular tissue, blood, lymph, interstitial fluid. You have blood. Other organisms like insects have a combination of interstitial fluid and blood, which would be called hemolymph. So there's lots of different fluid types in different organisms. Muscle tissue, pretty simple. We've got 3 different types in vertebrates: skeletal muscle, cardiac muscle, and smooth muscle. Depending on the the organization of the and complexity of the body form of an animal, it may just have smooth muscle or skeletal muscle. Nervous tissue is for conducting signals throughout the body. Of course, your brain is made of nervous tissue, and all your nerves are. There's 2 basic cells: the neurons, which are the nerve cells, which actually conduct the signals, and glial cells, or neuroglia, which are supportive cells. You can see those on the bottom left here. You've got different types: oligodendrocytes, ependymal cells, astrocytes, Schwann and satellite cells. All these are surrounding neurons in different ways, make them conduct signals faster, or create barriers, or have other supportive functions. All right, finally we have extracellular components. There's extracellular fluids in closed vascular systems like blood, lymph, and interstitial fluid, which we talked about before. And there's also extracellular elements such as cartilage, bone, and cuticle, which provide mechanical stability and protection. All right, last thing we're going to talk about is body size and complexity. So unicellular organisms have advantages in that they don't have to worry about getting energy and nutrients and waste products very far because they're so small. So it's very easy for them to exchange with their environment. However, they're very limited on their ability to acquire resources, acquire energy, and move through their environment. So by increasing in size, you can better handle environmental fluctuations just by your size, whether it's against changes in heat or defense against other organisms. So that provides you protection. You can move a lot better, and you're more efficient in resource use. Yes, an elephant uses a lot more energy than a mouse, but per unit area, it's much more efficient. So overall, or per cell, per kilogram, an elephant's going to use a lot less energy than a mouse. But you do have to deal with the problem of increased volume. So the larger you get, the harder it's going to be to get the nutrients and things you need to your cells. And so the multicellular organism solves that problem by creating lots of little cells, which increases the surface area. Cells don't get very big for that very reason. You can't get all the nutrients and waste products in and out of a very large cell very efficiently. So we're going to just make a bunch of little cells, and then we can have a method of delivery for all these little cells in a circulatory system so that the necessary resources and the waste products can transfer efficiently in and out of those cells. And there you have it.